Yes, Dr. Perlingeiro is the Associate Professor of Medicine at the Lillehei Heart Institute at the University of Minnesota. Her primary research interests include understanding the molecular mechanisms controlling lineage. We're talking about cell lineage, like when you start with an embryo, how does it become just an embryonic cell into skin, muscle, all the different parts that make up our body? It's a very fascinating biological question. And she's looking at how these things that you'll learn about called pluripotent stem cells, how they determine their fate and become different types of cells. And then how do you apply this information to generate tissue-specific stem/progenitor cells that are endowed with in vivo regenerative potential. And her work has potential applications in muscular dystrophy research and future therapies. So I will turn it over to Dr. Perlingeiro to explain a lot of these terms to us and concepts. Welcome.
Thanks, June. Thanks, the FSHD Society for having me today. It's always a pleasure to talk about our work and discuss how do you begin and where we stand now. So for questions, for a point of time, I will try to go a little bit more directly to the point, not too much of the history. I reduced those slides to discuss more the recent work, but I'm happy to answer any basic questions, anything that comes to your mind that I can answer in the end of the session, if there is limited time, by email. Okay, always happy to answer questions. So as June said, I'm professor here at the University of Minnesota. I have been here for the last 10 years. I was at UT Southwestern Medical Center in Dallas before. And since the beginning of my independent career, I have been interested in muscle regeneration from pluripotent stem cells. My training was in George Daly's lab at the Whitehead Institute in Boston, really focusing on blood from pluripotent stem cells. So that was my entrance to the pluripotent stem cell field. And pluripotent stem cells are a very interesting type of cells because they are very plastic, if you will. They have the potential to generate different cell types. And when we began in this area, focusing on the skeletal muscle, there was not really anything there. So it kind of, we can say our lab was a pioneer in this field, and I'm glad to see that lots of different labs are working on this area because there is just lots to do. So this slide is to give a sense of the potential of the cells. So just a brief explanation on what stem cells. There's a lot of different misconception, right? Or a lot of discussion on stem cells and what different types of stem cells. So let's divide it simply into types: adult and embryonic, or more immature or pluripotent stem cells. Adult cells, stem cells, they have been identified in the skeletal muscle, in the blood system, and they are able to differentiate it only in one cell type. Okay, so in the muscle stem cells can only make skeletal muscle. The blood, the hematopoietic stem cells can only make blood. They are great, they know very well what to do. One of the limitations of the cells is that it should obtain enough, in particular in the skeletal muscle lineage, because you need to obtain large number of the cells for a transplant. So having enough, because they are so special and they know so well what to do, you don't need lots of them in the body. But if you're gonna re-isolate those cells and perform a transplantation, then you need lots. And what happens is when you put the cells in the dish to make lots of them, any adult stem cell tend to differentiate. Even though, you know, we consider ourselves very smart, the scientists, you know, the cells seem to be much smarter than us, and somehow they have their own wish that they don't want to keep in that state of stem cell. And a stem cell, just backing up a little bit, it has the properties of the stem cell. Is a cell that has the capacity to make more of themselves, so proliferate, and differentiating the cell that you want, a functional tissue. talking here about muscle. So this is the limitation of adult stem cells. They're great because they are very specific. They know what they want to do and they do it well, but the number is a problem. Okay, so getting enough of these cells. And because they differentiate when you put in the dish, is also difficult to manipulate these cells. And when I say manipulate, is to engineer them, for example, to correct a mutation. Okay, and when they start to differentiate, they lose their stem cell properties. And if they lose their stem cell properties, they also lose their regenerative potential. So that's the conundrum in the field of adult stem cells. So alternative to adult stem cells is the use of pluripotent stem cells. So this is a cell type that is less specific. So it was first identified by isolating cells from in the mouse, from the inner cell mass. So first work was done with embryonic stem cells that you might have heard about it. And these cells, they are very plastic. And as you'll see, the work started with embryonic stem cells, but nowadays we all kind of switch to induced pluripotent stem cells. So I will introduce that concept later. But for now, let's just think about pluripotent stem cells. What are these cells that are not adult? So these are cells that can be maintained in the dish indefinitely. They're very immature and they are amenable to genetic modification. So there is no issue anymore on generating very large numbers of cells. And a very nice aspect of these cells is that they can differentiate in all different cell types. So they are, and that's actually what defines this as pluripotent, right? So pluri has the capacity to generate multiple cell types. So if you differentiate it, if you put the cells in the dish, here it says IPS, so pluripotent stem cells, they are able to differentiate in different cell types. So this is very exciting because it allows you, if you have a controlled differentiation process, you can make neurons, you can make skeletal muscle, pancreatic islands, cardiomyocytes, and these cells from the patient can be used for studying the disease, can be used for drug screening. Obviously, we are very excited in our lab is to generate these cells for regenerative purposes. And when you're discussing genetic diseases that are associated with genetic mutations, you can think about doing the gene correction of the mutation and then generating the cell types and putting back into the patient. I'm gonna get back to that. This is just a more kind of general slide with the idea that these cells have the potential to generate cell types. So everything is beautiful and exciting about pluripotent stem cells? No, there are limitations as everything, right? So in one side, it's very exciting because you can generate all the cell types. In the other side, because they can make every cell type, you wanna make sure you develop a particular protocol that generates just the cells you are interested. For example, for us, you are interested in treating muscular dystrophies. So our cultures, we don't want neurons or you don't want any blood cells, right? So the very guided, instructed differentiation is very important. Another point is you wanna make sure that you have a pure population. There are other caveats there, but we'll go as we discuss. So we are focused in skeletal muscle in my lab. We have been studying this, we have been in this field since 2005. And more recently, we are focused not only on the regeneration with the advent of iPS cells, but also using cells, this material to study disease and drug screening. But I would say that this is really the major interest in my lab. So there is a potential for in vivo regenerative medicine, development of cellular therapeutic, disease modeling for drug screening. So as you would imagine, this there has been already in clinical trials for some diseases, and two particular diseases that iPS cells have moved to the clinic or are in clinical trials is macular degeneration and Parkinson's disease. These are here the groups that have been focusing on these treatments, but there are multiple around the world. And one aspect here that I would like to emphasize that It's never easy. Nothing is easy. But one aspect that I'd like to point out is this particular disease is that it's a very specific cell type that needs to be generated. There is no really degeneration going on in the eye or in Parkinson's disease. So as long as you provide functional tissue, the final cell that you need, and in the right place, which is not large, the results are very encouraging. In skeletal muscle, as you already can imagine, the challenge is a bit steeper because we needed to provide functional muscle, but you also need to provide stem cells, and we're gonna get there. So, as you know, muscular dystrophies also are not diseases that are very localized to one small area, right? So, it's a very heterogeneous group of diseases, several types of muscular dystrophies, and within the several types, several different types of mutations. So I don't have to go on and on that. We have this cartoon exemplify different types, including FSHD here, limb girdle, Duchenne, and Becker. And in my lab, as you go through, you'll see through some representative slides, we have spent significant amount of time working on Duchenne. You also have projects on limb girdle, and you also have a project in the last 2 years on FSHD. So you've been exploring the potential of pluripotent stem cell therapies for this disease. And how do you do that is really using animal models of the disease when available, so we can test the therapeutic potential in these different types of the muscular dystrophies. But going back, well, very clearly, we need to reach lots of muscles, right? But we're starting small, so we focus on one particular muscle at a time. So this is very different from these other diseases that have been are in clinical trials now. And another very important point is because of course, if you want to generate cells for therapy, you have to think, what do I need to treat this disease? Obviously, you need skeletal muscle, right? But in this cartoon, I just want, in these images, I just want to point out, this is an example of the progression for Duchenne muscular dystrophy. Of course, every muscular dystrophy is a bit different, but overall, at the end, you have muscle wasting. And muscle wasting is happening because different mutations will lead to muscle degeneration of the myofibers. There are these fibers here that you see. I don't have the example of a healthy muscle here, but this is early stage. So what I want to point out here, these are the healthy myofibers, and then you start already seeing signs of inflammation and fibrotic tissue. Okay, so by the end in Duchenne, you can see you don't really have these beautiful rounded myofibers anymore. So it's totally fibrotic and with fat deposition. What I want to point out here is that when you have a myofiber that has a central nucleated, like this one, you have lots of myofibers here, it means there are newly regenerated myofibers. And this is the indication actually that we have, remember I mentioned in the beginning, you have stem cells in the muscle, they're called satellite cells. And these cells respond every time there is injury, just because you exercise a lot or because you have a chronic condition, your stem cells will become activated and will attempt to make new muscle fibers, and they do. The issue is that depending on the different pathologies, these new myofibers will degenerate because of the mutation. So the stem cells work pretty hard, and this process of degeneration and regeneration is constant. So thinking about providing or making a cell therapy for muscle diseases, in particular muscular dystrophies, is very clear that the cell replacement, we needed to target the myofibers. Well, these guys here that actually will are responsible for the contraction, the muscle, a healthy muscle. But we also needed to provide the satellite cells or the stem cells, because these are the cells that will be in the muscle long-term. And when that process continues of degeneration, regeneration, the idea is that those healthy transplanted stem cells will be there long term and keep contributing to muscle regeneration. So that has been our goal from the beginning. Again, different from macular degeneration or Parkinson, that you just need the final product, the final functional differentiated cells. In the case of skeletal muscle, you need to provide both, because that's the only way to provide long-term regeneration, okay? So I said not everything is perfect and easy, right? So in science, usually we have always, even though when something is very exciting and there is a lot of potential, things are challenging. And the making a specific cell types from pluripotent stem cells, although we have learned a lot in the last decade, there are still challenges. How do you make a particular cell type from this mixture of cells? So this is just an example to show. This is mouse embryonic stem cells. This is one of my first slides. This just shows the cells here when they are undifferentiated, mouse cells. And when you allow the cells to differentiate in the dish, they make these balls, what they call embryo bodies. And they're very tiny, as you can see. And by 1 week, they become these big balls. And we call embryo bodies just because they try to mimic an embryo. They are not an embryo, of course, they are not organized. But you have cell types inside this embryo body. And the first evidence is actually embryo body has differentiation going on. Can you see these red spots? So these are actually red cells. And if you keep looking at the beads, you can see they beat. So not that they are full of cardiac cells, but there are some cardiomyocytes, and that's enough for us to see the beating, which is always still at this point get us excited in the lab. So this was the first evidence that actually these cells are able to generate different cell types in the dish. But as I said in the beginning, as you can imagine, if you don't instruct the cells to make something, a cell type in particular, there's quite a heterogeneous population. Depending what you add to the median, to the growth conditions, you have, more or less of different cell types. And you also have residual undifferentiated cells in the dish. So the point of this slide is to emphasize that despite this is a very exciting population to explore for therapeutic purposes, it's critical, it's very critical that you guide the cells specific to the cell type you want, and you target the cells you want. And here again, we are interested in generating cells that will give rise to muscle fibers and also able to make stem cells. So you want to really target a very early population of muscle progenitors, as we're going to hear me say during this talk. And we also needed to target a population that you also can make lots of them, right? Because as I said in the beginning, if you're planning for transplantation experiments, you need millions of cells, right? So you want to make sure that those millions, billions of cells, they are as homogeneous as possible and muscle. So how did you do that? So from the beginning, this is Rita Perlingeiro, that was my postdoc back in Dallas that led this work. And the idea from the beginning, based on my experience in the blood field too, was to try to target, because we knew already from early work we had done that indeed it was a very mixed population and there was not much muscle going on in those balls. So we decided if you want to really induce the skeletal muscle fate, we went back to development, how muscle is formed, and you took advantage of these two factors here, these genes, PAX3 or PAX7. They are very important during development to instruct the cells to make skeletal muscle. So they are really on the top of the hierarchy, if you will, of a muscle to tell, you guys, you don't go pancreas, you go muscle. Okay, and these markers or these genes, they are expressed in very early progenitor cells for the muscle fate, and in particular PAX7 is a marker of adult muscle stem cells. So very clearly from the beginning for us was obvious that these were the genes to target. Okay, so I'll just explain here and then I will focus more on the transplantation data. But the idea here is to make from this pluripotent stem cells lots of skeletal muscle, we introduce one of these genes in these cells in a controlled manner. So you turn the gene on by adding doxycycline, which is an antibiotic. So this is a trick to just generate these very large numbers of progenitors in the dish. And once you do that, you're going to hear me again say PEX myogenic progenitors, you generate very large of this population that grows in the dish in response to doxycycline and some growth factors that muscle progenitors like. This allows us to generate very large numbers of these progenitors in the dish, and you're going to see some examples of this growth later on. And if you remove this DOX signal, the cells differentiate into myotubes, into muscle fibers in the dish. And if you transplant them in animal models of muscular dystrophy, we have done a lot of the early work again in the dystrophin deficient animal model because it lacks dystrophin, so it's very easy to visualize, but we have been doing this for different animal models for different diseases, including FSHD. So here is an example of engraftment of muscle fiber, and this is an example of satellite cell engraftment. As I mentioned to you in the beginning, you want both. You want the cells that you inject be able to make muscle fibers and stem cells. And this is an example of the muscle stem cells. They express PEX7, and they are called satellite cells because they like to be in the satellite cell position of the muscle fiber under the basal lamina. So there are ways of identifying that. So here is one single myofiber that's green, it's green fluorescent protein positive because our cells were labeled, and you look at that in this particular location, okay? Of course, you wanted the cells to make myofibers and make stem cells, but in the end of the day, what do you need? You need force, right? So are the cells you transplant, these cells use myofibers you see here, are they doing anything, right? Because that's an important question. Sometimes you might see cells sporadically in different areas, but they are not providing functional contribution. And we have done this in several ways, and we isolate the muscle that was transplanted, actually intramuscular or systemically, we have done in mouse-to-mouse work, and you see an improvement in contraction. And this is what you are measuring here, specific force, okay? So as this slide that I show was just with mouse ES cells, and you might like, I know that there's a lot of controversy with embryonic stem cells for good reason. And when we began this work, there was a lot of questions. Yes, this is great, but how this is going to be translatable to treating diseases, right? Well, you're lucky enough that while you're doing our work putting PEX3 and PEX7 in mouse ES cells, Shinya Yamanaka in Japan was actually advancing big discoveries in how to generate pluripotent stem cells from adult cells. As I told you, pluripotent stem cells, the first identified was the embryonic cells that you have to collect from embryo. So obviously not very useful for treatments, right, without ethical concerns. So Yamanaka came with developed a strategy for which he won the Nobel Prize a couple of years ago, in which he discovered a strategy to reprogram adult cells, any cell type, into a pluripotent stage. So to that stage like embryonic cells. So what is the magical bullet? So through many years of work, he identified that these particular genes, we are working on PEX3 or PEX7 to make muscle. So he identified these other genes, if introduced to any cell type in the body, even fibroblasts. And when you talk about fibroblasts, it's like a skin biopsy, like just skin cells that cannot do anything, right? If you introduce this skin fibroblasts with these genes and culture them in the same conditions as embryonic stem cells, they become pluripotent. And that's what you call induced, right? They are not pluripotent per se, but if you introduce these factors, they can become pluripotent. And this has been extensively characterized. And as you can imagine, when it was first reported, a lot of people, is this really real or is it robust enough? And it is, because if you do a quick Google search, you're going to see there are more than 5,000 papers on this field. Well, probably more than that. We have been working on this since we moved to Minnesota for 10 years, since the beginning. So just to give you an idea of this magic happening in the dish, because I still find that really remarkable. So this is how fibroblasts looking in the dish, skin cells, right? So they look elongated, and that's just fibroblasts. Then you transduce these factors. You do a genetic manipulation, you introduce these factors, and 2 days later, you already can see a little bit of difference. A lot of cells don't like it. Some might die, and others just stay the same. They don't want to change. But some that receive this factor, they start to change. And you can already see these changes after 10 days. In 2 weeks, you start seeing these colonies forming, which keep growing. And after 3-4 weeks, it's very clear that we have these colonies that look like pluripotent stem cells. So what we do, you pick the single colonies, and you expand and establish pluripotent stem cells. So you can imagine the revolution in medicine, right? I started with that slide that gave away already what you can do, but you can now generate pluripotent stem cells from anybody, and in a non-invasive manner, independent of age, independent of disease or not. And really allowing us, if you have guided differentiation protocols, to generate specific cell types, whatever you want, even cell types that sometimes are very hard to obtain, like heart and neurons. So we validated this using several pluripotent stem cells from donors. So this is a normal, non-affected. This was the principle, proof of principle work. It was published several years ago, but it's just for you to see. Let's see if I keep track of time. I'll pick up the pace here. So this is cells, how they go through the differentiation. At this early work, you isolate based on green fluorescent protein, which isolates the progenitors that I told you about, that's the cells you're interested. You can grow lots of these cells. These are the PAX7 myogenic progenitors, human, okay? And in the presence of that doxycycline, we can grow, this is just an example how much they grow. It's exponentially. So and we can start this with very large numbers. There is no limitation of how many of these you can get. So number is no longer an issue. These cells differentiate in the dish. These are 2 examples of iPS cells, induced pluripotent stem cells. We transplanted these cells in an animal model of Duchenne. Here we are transplanting human cells into a mouse. So we use an animal model that doesn't have the immune system, so we can easily look at the engraftment, the presence of donor cells. So here you're looking for human specific markers. And then as you can see here, you look for several markers to make sure that what you see is real. So here is a human specific marker for the nuclei, human lamin AC. You can see control muscle that was injected with UBS doesn't have any signal for dystrophin or lamin AC. And you see muscles transplanted, you can see myofibers that are human donor derived. Similar engraftment, we see improvement in contraction. And again, we see the presence of stem cells, donor-derived stem cells in the right location. Okay, so we hit the 3 areas: muscle fibers, enhanced muscle function, and the presence of muscle stem cells derived from human myogenic progenitors. And they are there long-term. That's always a key aspect, right? So even though they do seed the stem cell pool, they are in that location, so they should be there long-term. The question is like, are they really, right? Because you have to remember that our stem cells are there. So there is some level of competition there. And the idea is that over time, the healthy cells will take over. But this is something we can only test in the animal model at the moment. You know, we will really know this for real when you get into patients. But another point that we transplant and waited for a long time was not actually just looking for long-term graft, but you want to make sure the cells are safe. So as you can see, this is after almost a year of transplantation. You can see we saw no abnormal growth, no abnormal or presence of cells in different tissues, and we just saw skeletal muscle, and the engraftment was persistent. So of course, these mice were not subjected to challenges in this particular study. You just look at them later on. So when you talk about, I'm going to get more targeted here in terms of where we are and we have lots of very basic research projects, but I'm gonna focus on our recent efforts and where we would go to translate, right? I think it's always a dream of a scientist. Can I apply all this work we are doing in the lab to change the lives of patients, right? So that's always a dream when you start your PhD. And this is still my dream and the dream of my lab members. So when you think about transplantation, right, very generic, this slide, you think in two types. Allogeneic, when you have one cell population derived from one donor, and this donor doesn't have any disease or not the disease that you're thinking to cure. And these cells are used to treat lots of recipients, right? And doesn't matter the sex, doesn't matter the age, and you can use the cells. So for example, allogeneic is used commonly for bone marrow transplant, right? So most of the time you just look for compatibility, which you probably have heard about it, but I'm not gonna go into details of that today because of time. But that's the idea. You have a healthy donor and you generate cells that can be used in lots of people, okay? In our case, we can think of that using with the cells you have generated. You generate lots of healthy cells, you bank them in the freezer, and then you can use for a number of patients. Of course, with iPS cells, there is a lot of excitement with autologous because now you're really being specific, right? You generated cells from one patient, and then you treated that patient, and you don't think too much about rejection, at least, right? Immunohistocompatibility. But when you're discussing genetic diseases, of course, we have to think about correcting the mutation, right? So that simple slide gets into these more complex slides. Bear with me. So if you're going to think allogeneic, you generate the iPS, you reprogram those iPS, you make those muscle progenitors with PAC7, and then you transplant into the patient, right? This will be the allogenic route. The autologous route, you get the iPS cells from the patient, you get fibroblasts or any cell type from the patient, reprogram them, generate disease, the patient-specific iPS cells. Of course, if it's a genetic disease, which is the ones we are focusing, we need to correct the mutation, generate the progenitors, and in theory, you can transplant this back, right? Super exciting. So before I'm gonna talk a little bit more about our more recent translation efforts, I'm gonna show you a few slides showing the proof of principle that this can be achieved, right? So for this project, we have a couple of projects focused on gene editing of patient-specific iPS cells. This one was limb-girdle muscular dystrophy 2A, which is mutations in the calpain 3 gene. So we have cells from different patients, different mutations. I'm not gonna tire you out with this different disease. So we have these different lines. We differentiated these iPS cells from these different patients' disease samples, and they make myotubes in the dish, as you can see. And then we look for protein expression of this gene that's mutated. So as you can see, a control line, you can detect calpain 3. And the 3 lines from the patients we cannot detect. And this lack of calpain 3 is that leads to the muscular dystrophy in this patient. So a very talented graduate student in the lab generated a gene correction strategy that you cut the genomic DNA of CRISPR-Cas9 and you come with a donor vector. Again, I know this might be over the top for you, but you introduce the cDNA with the normal sequence of calpain 3. Okay, so a lot of steps, molecular biology steps, and you select clones that are corrected. Okay, so there are several steps that you go through. Making a very long story short, after we screen for several clones, then the end point is like, can you rescue that protein for calpain that was not there, right? So I circle here for you. So these are the 3 lines of patients we had. U stands for uncorrected. As you can see, there is no calpain 3 here. And then C1, C10 is corrected clones. C5, C10, C1, C3 are corrected clones for each line. And you can see there is rescue of the protein expression for calpain 3, which is mutated. So the question is this really functional, right? So we transplanted animals that don't have calpain 3, and we transplanted these mice with these 2 clones, and you can see there are a lot of donor-derived myofibers here. can really detect kappa in the enzyme. So it's very hard to see by images. So we collect the muscles and you can see human kappa-in3 expression only in the muscles that were transplanted with the cells, nothing in the control. So this shows that this is possible, right? We can do this. We have done for Limb-Girdle 2A and we are doing for different diseases. So, okay, so let's transplant patients. Well, not so fast, right? Because when you start thinking about, okay, we have done all this work. Well, you know, I get this question a lot from families. So what do you need to get this into patients? Oh, you know, this can take my sleep at night because, you know, what do you really need, right? So there's just so many questions. Even though we feel we have accomplished a lot in the last several years, there were still lots of questions to answer. But one of the main issues that I pointed out here is like, well, if you're going to do for clinical application, not mice, well, you need to really make sure that the manufacturer is compatible for patient application, that we have a very good and doable purification strategy. You have to make sure that you can scale up, right? Because we know we can, but we actually never done billions and billions because we don't need to, right? Safety, and of course, later on delivery, you know, right now we are focusing on intramuscular and we have done systemic, but you know, delivery is always a big question for muscle diseases. So as you can see, you know, you can see how each one of these are quite complex. And we thought that going with an autologous route, when you have one cell product that can be used for one patient, probably wouldn't be the best way for us to get started, even though it might be the most exciting. So we decided that you would focus in the allogeneic. When you generate cells from a healthy donor, and you can really optimize the manufacture, the purification, do a lot of testing and characterization of the cell product. Now we are talking, you know, not talking about cell product because you're thinking about a cell that actually can be used for treatment. And you want to make sure when you do the safety that you can do actually in a couple of patients and not in one, right? Because it's very hard to do safety study in one patient, right? And not talking about all the cost associated with this process. So how to begin, right? So this is also a very brief cartoon, how the roadmap to clinics. I mean, this is very simplified, but you're pretty much here, right? We had done a lot of research and we had done proof of concept with autologous and allogeneic, but how do you get here, right? So there are all these different expertise that you need that you might not have. We have the scientific expertise, but not necessarily the production expertise and testing and moving to a clinical trial. So this question, how do you get there, started back in 2016. Prompted by patients, I have to say. Not that we didn't want to get there, but you know, that is fear associated with all the challenges of the journey that I knew there were lots of them. But if you are with the right team, if you assemble the right team, we felt that we could do this. So the group is John Wagner, pediatrics, lots of experience, cell therapy. Peter Karashansky, neurologist. David McKean is director of molecular cellular therapeutic, so he has a lot of experience with GMP, generating cells for therapeutic purposes. Robert Schumacher, Bruce Blazer, and Michael Kaiba, which you might have heard has been working on FSHD for a long time. So we have this facility in the University of Minnesota where you actually can produce cells for therapy. They're one of the references in the country for different cell types, not for iPS cells, but we have what it takes to do it. So, okay, so back in 2006, let's start tracing a roadmap for this. So this has been going since then, and this is a very short list of all the things that we needed to do from the proof of concept study. Methodology, purification, generate a GMP compliant cell product, make sure that this is scalable, characterize the cell product. Well, we have hit all the buttons in the last 4 years, and we are really in this moment here where you are transferring the technology to production and getting all the reagents to generate the product and generate the preclinical study. As you probably saw, we have done lots of preclinical studies in the last 6, 7 years. But when you want to test a particular cell for treating patients, you want to make sure, or it's going to be easier with the FDA if you actually do the work with the cells, exactly the same cells that are going to be injecting the patients, right? So that's what you're going to be doing, hopefully, in the next few months. Because of time, I will pass this, but this is just to show that we have tested different strategies and they don't work. So we are pretty set. I'll just pass this. We are pretty sad that we need to use PAC7 introduction to generate large number of cells that actually do engraft. We have tested different conditions, different protocols published, and they just don't work. Not for this purpose, not for regeneration. They do work for generating myotubes in the dish. Okay, I'll just pass because of time. If you use PAC7, we do see engraftment, as you can see here. And a very important aspect for getting our cells close to the clinics was to find strategies to purify, because I showed we purify based on green fluorescent protein, but we don't want to introduce green fluorescent protein to patients. So we want to identify surface markers that you can isolate these progenitors. So Alessandro Magli has done that in the lab. This work has been published. So we use antibodies. So we can tag the cells in this mixed population and get rid of what's not skeletal muscle. So we have identified a couple of surface markers, and we're pretty interested in this guy here, CD54. Just to show you that we are able to identify the population, and the triple positive is really mixed muscle, okay? We have transplanted the cells, and this is our control, the green fluorescent protein cells, and these are cells purified with the surface markers, and they behave beautifully, the same as our original protocol. When we were talking about this with our translational group, great, we have 3 markers, right? And then someone said, can you use 1 marker? And it's like, why? 3 should be better, right? Well, not. That is not better because it's more expensive. And when you're talking about bringing this to the clinic, you definitely want to make it efficient and I wouldn't say cheap, but less expensive as possible. And also be able to use not FACS purification, but this guy here using column, because it's a closed system that you don't want exposure of the cells to the environment or different cell types. So this is the ideal purification strategy used for different cell therapies. So we tested CD54, that's why I said it was our favorite, and it worked pretty nicely. So this is our strategy for purification of large scale myogenic progenitors for therapeutic applications. One of the questions you always have, if there are negatives in your positive population, what the cells are going to do? Are they going to make tumors, right? This is, nobody likes to hear this word, but that's always a worry. You want to make sure that you don't have any residual pluripotent stem cells in your cultures or cells that will make. So there are 2 points there. Is the safety. You don't want any cell type that's undifferentiated. And then also the point that you want a very pure population because it's going to be more efficient to make muscle, right? So if you transplant undifferentiated cells, they're going to make tumors, as you can see here. These are cells pluripotent in this undifferentiated stage. Didn't go through the whole process of making muscle, okay? So what we did, this was our control. So we look at the cells, the negatives and the positives, and they don't express any of the pluripotent markers after we go through the process. But how do you know for sure? Right? So you injected positive cells, our target myogenic progenitors, and the negative cells, and waited. And we injected excess of cells. So usually it's about 1 million, 1.5 million. We inject 5 million cells, and you wait for over a year, and we didn't see any tumor formation. So we are pretty confident that the cells generated in the cultures are pretty free of undifferentiated cells, but of course, all this study is going to be repeated with the cell product. And because we know you need the 3rd generation, we need the lentiviral vectors to make the cells, we are making use of 3rd generation lentiviral vectors to introduce PAC7 to the cells, because these are actually used in gene therapy trials, and they are considered safe because they are 3rd generation. Okay, so we moved all our system to these new vectors. This is the idea where we stand. So we generate allogeneic donor iPS cells that are healthy, and they have been extremely well characterized. We are using cells generated by NIH. We exactly for this purpose of having multiple investigators using for different purposes. We use third generation lentiviral vector that's being prepared clinical grade. Everything has to be clinical grade, okay? So that is the big step. And we generate a master cell bank of these PAC7 iPS cells. And then you do a whole genome characterization because you want to make sure we isolate clones that are safe. When I say safe, is that they don't have any integration or dangerous integration. So when you pick these clones, we go through the procedure and we can freeze large number of the cells. And you're going to do the preclinical work, and the cells hopefully will be ready for clinical trials. So where are we here? We are here. I've been seeing this for a while, but you know, it takes quite a while to go from here to here. So we have validated and optimized our strategy, all the steps, the scalability, the culture conditions, the harvesting, the purification. So we're really now generating the clinical grade vectors. It's being generated in a different lab. And we are pretty much very ready to start the process at the clinical grade facility. Once we have the product, then we're going to be doing the preclinical work and hopefully doing the first in human safety trial. The first goal is really safety, right? First, I guess one of the questions is like, can we manufacture the cells? And then the second question, if you can, are they safe, right? And then we go into a phase 2, phase 1B2. Because we had so many questions during the process, we decided to have a pre-IND with the FDA very early on. And for those of you that are not familiar, it's just a conversation with the FDA, with the Food and Drug Administration, to ask questions, to present your plan, how you envision this strategy being moved to a clinical trial, and if they're in accordance with our plan, if there are anything. So you have a kind of a roadmap, really. That now is a true roadmap of what the FDA is going to ask. Doesn't mean that they're going to ask the same. They might come up with different questions. Even our plan evolved over the last 3 years, but it gives you a pretty good sense. It's really a starting point. So we had that in 2017, and we have been using that discussion as our reference. And we have done the work, as I said. We have the NIH IPS cell lines, just to show you. This is how they look like. We introduced them with the third generation vectors. We use a large scale. Clinimax is actually the machine that people use for generating clinical grade cells for therapy. So we are able to adapt all our procedures to this. And as you can see, starting with 100,000 cells only, we get to 10 to the 10. So we get really easily billions of cells. We have the cells differentiated in the dish, and they are able to generate myofibers. So, you know, you have to hit all the marks, right? And so this is show you also an area of engraftment. Okay, so that's where we are in terms of moving this strategy for clinical trial. And you might be wondering where, who, right? This is still early for me to tell you, but I can answer questions where we are right now. And then the way I envision this trial, you know, in a small group of patient safety, and this is very important that you are very careful in all the different steps, because you want to be successful, because if you plan right and you are successful, this means that we can be using, that there is potential for using the cells for different diseases, different patients, right? So that's the beauty of the allogeneic, because one cell type, or let's say 3 healthy cell types that we generated and banked and characterized, could be used for a large number of patients, right? While the autologous, very exciting, it's going to take longer. And as you can imagine, the cost of generating one cell product per patient, right? So it is the most exciting path, but we can hit many more patients. We can treat many more patients if we go with the allogeneic route, if that works. But in any case, the allogeneic is the starting point. It doesn't mean that you would be, if it works, it also opens the doors for the autologous. So those different strategies can be going on simultaneously. So you might wonder, how about FSHD, right? So as I mentioned in the beginning, we started working on FSHD about 2 years ago, I think. And we're very fortunate to have funding from the FSHD Society to begin testing proof of principle for pluripotent stem cell-based therapy to rescue the phenotype. And as I mentioned, how do you measure phenotype? How do you know if the cells will be helpful in this disease? So the way to do it, we need an animal model. As you know, it was a while to get an animal model for FSHD. We have collaborated with Michael Kaiba, who has generated, DUX4, right? I don't need to introduce that. So he has generated an animal model with conditional expression of DUX4, which seems to be, I think is pretty consensus, that's the culprit gene for FSHD. So he has generated this animal model where you have DUX4 expression only in muscle. And he has published this recently. So he is the one, we are collaborating with him, and he has been providing these mice for us to test our cell therapy. Okay, so this is from his work, from his published paper. So here again, we use doxycycline. So when you treat the animals with doxycycline, DUX4 gets expressed. And then you can see the muscle is smaller than the control mouse. This is a control muscle biopsy. You can see very nice organized muscle fibers. I failed to show that in the beginning. And this is the muscle biopsy from this mouse after exposure for 6 months to DUX4. And you might know it's tricky to detect DUX4 in control mice. You don't see DUX4. Here again is the muscle fibers. And with exposure to DUX, you see DUX4 in the muscles. And as you can see here, that's the pathology, right? This is his work. We want to use these mice, right? So you need the muscle phenotype. So that's the title of our project. And I had Karin. Karin is working in this project. She's a postdoc in the lab. He has lots of experience transplanting different animal models. And every time we start studying a new disease, I have to tell you, it's a new challenge, because every disease has their own challenges and ways to learn about the model as well, not just about the disease, but a particular model. So we have been trying different strategies. As I mentioned, the muscle pathology of DUX4 gets expressed in this mouse if you treat with doxycycline. Problem, if you keep treating with doxycycline, this is also affecting our cells. So you have to trick the system. So we play, this slide you're seeing here is that you injure the muscle, or you expose the muscle to DUX4 by treating them with doxycycline. And you have here, I'm showing examples that you treated for 7 days and then did the transplantation. We also like to injure the muscle a little bit further with cardiotoxin because you see more. And this is something we do with other mouse models. This is something you need in the mouse model. And you also transplanted, and we had another cohort that you kept treating, giving dox to see if keeping the DUX4 on would have a difference, right? So this is just the examples. We can see the cells doing graft. This is mouse cells into mouse because we don't have an immunodeficient animal model for FSHD at the moment. So here, this is pure proof of concept, okay? So we can see in white are the muscle fibers, the donor cells are red here, and the nuclei. So here, all the areas that you see red and white, these are donor-derived myofibers, okay? So the cells definitely engraft in this animal model. This is lower magnification, it's hard to see, I think, so I would focus here, but this just gives a sense of the area, right? And yeah, let me see, where's my mouse? I think I lost my mouse. But you can see here, okay, the quantification. So you can see that if you keep dox, you might see more myofibers because you're still giving the signal of DUX4 and the signal for regeneration. So it's definitely clear that the pluripotent stem cells, the myogenic proteins, are able to engraft in these muscles. And I was just talking to Karin this morning, we have just now been able to analyze a new cohort because as you can imagine, with the shutdown, everything was reduced, minimal capability, but you are able to do a new transplant 4 weeks ago. So he's ready to analyze. And we did something interesting in this new experiment is that here we treated with, we exposed DUX4 and you had to stop and transplant and wait, right? So there is no DUX4 after that. But in this new experiment, we actually treat, transplant, and give another boost of exposure to DUX4 in the last week. And you want to see if the engraftment is still persistent and how do the cells do in the context of DUX4 longer term. So we're excited about this work. So hopefully we are hoping to wrap up at least this story in the mouse to mouse, have this proof of concept, hopefully being able to do functional analysis in this mice as well. And hopefully work with Michael Cabot to generate a human immunodeficient animal model for this. But this definitely is the similar proof of concept we have done for other animal models. So it would make FSHD another disease to be eligible, let's say, for cell transplantation. So I would like to end here. Oh, I managed. Thanking my colleagues, my collaborators. I would start by actually thanking the group, right? I have the privilege to work with this amazing group of scientists. And I have circled here the players that I try already to show during the talk. But Alessandro and Jen are really the key players on helping moving our strategy to the clinics. Karin is our transplant or surgeon, if you will. And Sridhar, it was a graduate student working on the Calpain tree and also work on several aspects of transplantation. And also Tanya they both moved on. They are in different positions. She's in Sweden working for AstraZeneca. He's in Stanford doing his postdoc. So it's very rewarding seeing them go on in their careers. And the collaborators there, this is the collaboration, the translational group, different groups along the way, and of course the funding agency. It really takes a lot of effort and funding to do any work in this field. So I'll be happy to take questions. Thank you.
Thank you so much for rejoining us today. I'm sorry that our webinar got cut off previously, and I really appreciate that you're back here to answer questions from our audience. So I think the big question many people had is, when and how soon will stem cell trials be ready for people with FSHD?
Mm-hmm. Okay, well, that's a big question. You know, when you add the FSHD, so I can...
Oops, I'm sorry, God.
I can give you a sense that hopefully if everything goes well with the manufacturing that, as I mentioned in my talk, we have been working hard in the last several years. We are getting ready to produce the product, to generate the cell product to clinical grade. And then we're gonna do all the clinical, all the preclinical work, because you wanna do the preclinical work if the cells will be injected to the patient. So if everything goes well, and you always say with a big if, right? So far so good, but we never know when hiccups might happen. If everything goes well, we are hoping that you're gonna be starting filing for the IND sometime next year, probably summer or fall. So that is to start to have the, and if FDA likes, right, another if everything is okay, then probably 2022 or 2021. I don't really have a sense of how long does it take, but if you have submitted your materials to the FDA, usually they respond within a month. So from there, you know, it all depends if they have more requirements or not. So our goal is to provide as much as you can, so we don't have too much back and forth, but no one can really anticipate what's going to be asked. So hopefully early 2021, and we are focusing, at least at the moment, you know, this keeps evolving, but because of the high risk, it's still high risk in the sense that the first time, right? So I would like to remind everyone, this is a safety trial, right? We want to make sure that we can produce this, it's safe in the preclinical work, and it has efficacy. But in patients, our first question is this trial is really focused on checking if making sure the cells are safe. Yeah. And so it's the FDA So it makes sense for the FDA and for several people in the field that would go with a disease that every disease deserves a clinical trial. But the question is, which one is fatal or fatal faster?
Yeah.
Right? So I think there is a consensus in the field that Duchenne is a good disease to try.
Yeah.
And also another reason is that outcome, the measure is very clear. You want to detect dystrophy, right?
Mm-hmm.
As you can imagine, we go back and forward with the disease because we also like a disease or target a patient population that's not in the, you want obviously the disease to be, the muscle to be affected, so the cells are needed, but we also don't want a muscle that's totally fibrotic. And I think I allude to that in my talk because we are providing a biological product, right? So if there is very reduced oxygen, no blood vessels, the tissue is not, I wouldn't say healthy, but not with conditions for the cells to survive, our likelihood of success is very low. So we need to think very carefully about the targeting.
Right. And when you mentioned safety, what are the safety concerns for this type of therapy?
Mm-hmm. Oh, it's a cell therapy, right? So the cells will be there long term, hopefully.
Mm-hmm.
So first question that comes to mind, especially being pluripotent, if the cells will become anything else, and in particular, if they're going to make tumors. And that's not just for pluripotent stem cells, but any cell type that been growing in the dish, are they safe when injecting to a patient? So we have all the work suggesting it's safe, but you won't know until you do it, right? So, so this has to be proven. And for that, we are also planning a trial in accordance. And that's one of the reasons, and maybe I don't think I answered the question, but maybe I allude to it. And that's why it's intramuscular too, right? Because you don't want to put cells in the bloodstream, which are going to be spread around all your body. So this is another reason to be a local injection and in a muscle, in a muscle that we have easy access. By, we don't expect that, but in the worst case scenario, something abnormal happens, we will be removing the cells from it.
Right.
So you have to be prepared, right? We don't expect, but if that's something that happens, we'll be prepared to remove.
Right.
And other side effect, I mean, this is a cell transplantation, so immunosuppression is going to be required. So even if it was an autologous cell transplantation, you would require some level of immunosuppression. So we don't expect this would be permanent, but you will have immunosuppression for at least 2 months. So there are some effects associated with that.
That's interesting. Even for autologous, meaning cells from the patient's own body.
Right, right. So if you,
why is that you would need immunosuppression?
Right? So let's think about diseases that are lacking a gene, right? Different from FSHD. So patients with Duchenne don't have dystrophin, right? So in some might express some, so they might not be foreign protein. But in a case of patients that have zero dystrophin, which is many of them, they have never seen that protein, right? So you are putting the cells, even if are the cells from the patient that you engineer and express dystrophin, if you're putting those cells back, the patient's immune system never saw dystrophin in their life. So the response is to create antibodies against it. It's a different type of rejection, right? Or complexity. Of course, you won't have a cell rejection, but you might have issues of immunorejection or reactivity to dystrophin.
Right.
So it wouldn't be, again, permanent, but you would have for some time. And that's why gene therapy trials use immunosuppression. Not only because of the viral capsid, but also because of the gene.
I see. But in the case of FSHD, if you are shutting off, let's say you treat autologous cells to suppress DUX4 expression, that might be less of an issue.
Right.
in that sense.
Right.
Okay.
Which is a good thing.
Yeah. So now we get this question often too, whether their parents who are banking umbilical cord blood, hoping that in the future it might be a source of, stem cells or progenitor cells for future cell therapies. Is that a reasonable idea?
That's how I started in the field, actually, many years ago. Because when this family approached me back in Texas, they want to know about cord blood, because actually in between my postdoc and my independent career, I work in a cord blood company and we focus on different applications. So what I can tell you is that cord blood cells won't make any muscle. Okay, so they are very useful to treat hematological diseases. If you are talking about a different lineage, potentially, things related to blood vessels, because even though they are not really blood vessel parents, there is some exchange there, and there is some evidence that they might help. And there are lots of clinical trials for ischemia, but blood cells won't make muscle. So I can tell you that.
Okay.
One thing, and the same for bone marrow. Bone marrow cells don't make skeletal muscle, and I know there are trials out there. But what one could envision now, after all this progress, is that you could reprogram the cord blood cells to become iPS. And the iPS could be used to generate skeletal muscle.
I see. But there would be other potential sources for those types of cells that could be reprogrammed as well, right? So,
yeah, right.
So a number of years ago, we heard reports of efforts at muscle regeneration, for example, in war veterans who had major traumatic injuries. I think the Department of Defense was funding some of that research. And I recall a story about a kind of synthetic mesh that was implanted, and it was also permeated with, I think, autologous muscle stem cells, or supposedly, that would then hopefully regrow and repair that muscle. And is that a kind of approach that might have promise? And where has that line of research gone?
Yeah, so I'm not, I'm not familiar with clinical trials. I don't know if they have evolved to that, but I'm familiar with the efforts of the Department of Defense, because I think they even see the potential of what you're doing, because they do fund work on the Shen, and we have a grant from there. Actually, it was one of the initial funding we got to move this strategy to clinical trials. So the way I see this is really a combination. The stem cell biology for sure has a lot of potential for not only the chronic disease, the genetic diseases, but also these debilitating conditions from war or accidents. That would combine, I think the key there, we haven't been focusing on this area of investigation, but there is a lot of potential is combining stem cells and bioengineering. Because as you mentioned, we are thinking about recreating the muscle, and it's not just the cells, right? So you need the cells, you need also the other cell types that are important for the muscle, and you need material to put this together, right? So I'm familiar of different studies doing work in mice. There are people in Duke. I think they have done some work with using myoblasts. Again, and the idea is that if you have myoblasts, that's good. I mean, you can generate the muscle. It just would be important to have stem cells in there in addition to myoblasts, endothelial cells, and other cell types that are important in the muscle. But this is going, I'm not really familiar with recent work that's evolving to patients, but I know people are active working in this area, in the preclinical, in experimental work.
Right. So I have a question here about if, let's imagine there is an FSHD trial, regenerative therapy that is about to go into clinical trial. What do you think might be the inclusion criteria for that trial?
I don't really have a clear answer for that. we have discussed FSHD as one of the diseases you would like to target. And as I mentioned, once we prove safety of the cells, we envision opening up for different diseases, including FSHD, because we have even evidence that a cell therapy could work, you know, and I feel it's important to mention that it's like, oh, you're saying that works for every disease. You know, we have one particular type of muscular dystrophy that we didn't see an improvement, and that was myotonic dystrophy. And we published that. So we observed that the toxic RNA actually moved into the cells that were engrafted and healthy. So it's important to say that it's not, oh, okay, you're saying it cures everything. So, but we have evidence that for FSHD, it might have a positive impact. I don't really have an answer, because I would say I would have to discuss this with my clinician colleagues, but we would have to discuss that. But one of the aspects that you would be, you know, being able to receive immunosuppression, for example, is for sure one criteria for everyone at this moment, right? It's an allogeneic transplantation, and it's very important that for a few weeks the patients receive. So if there is any condition in the patient that they cannot receive immunosuppression, like being immunocompromised, that would be definitely a exclusion criteria. Right. Obviously, having a confirmation of the diagnosis would be an inclusion criteria. Other than that, really, I don't want to explain too much, because I don't really have a clear path at the moment. We are trying to establish that for the upcoming, and I would imagine that a lot of the parameters that are not specific to the disease will be the same for different types of muscular dystrophy.
Right. It's also, I think, important for people to understand that a clinical trial and who is excluded or included has nothing to do with who's eligible for treatment eventually if it's approved, because the clinical trial is an experiment, and you want to reduce the factors that can introduce noise and uncertainty in your data. So definitely, you have to match your trial participants to the types of outcome instruments you have. So if your outcome instrument is a walking test, then someone who's in a wheelchair would be excluded, not because there's anything wrong with, you know, that they couldn't benefit from the treatment, but they would not be able to participate in the outcome measure. Of the
Now that, I'm glad you brought that point. This is really more related to make it as clear and without noise as possible, right? Even knowing that even as much as you try to minimize that, every patient is going to be different, right? So to have as clear conclusions as possible.
And here's maybe our last question for this round, is would CRISPR play any part in the development of this type of therapy, for example?
In the autologous therapy, yes. So the disease I show you in my talk, that we correct the mutation, use CRISPR-Cas9 to insert the correct gene, to correct the gene, and you can use the same to correct or to remove, right? You can do different things. You cut and then you insert what we want, and you can cut to remove what you don't want, right? So definitely that's in the approach of using autologous cells. I know there are different labs using this strategy to correct FSHD IPS cells. That would be the way.
Great, thank you so much.
Collaborating and using these cells to generate an autologous.
Okay, thank you so much. So just to quickly reiterate, make sure I understood this correctly. The first muscular dystrophy that's likely to be go into a clinical trial for this type of therapy would be something like Duchenne. That's probably the most advanced, and if it's shown to be safe in that condition, then there's a likelihood that other muscular dystrophies, including FSHD, would be candidates for future clinical trials, but that's sort of the time sequence.
That's how you're planning to present to the FDA.
Yeah.
We see that they have, you know, of this idea, or presenting as an umbrella, right? That in particular for this disease, that's not a gene therapy, that's specific for a particular gene that can be used, if it's safe, can be used for different muscular dystrophies. And I think that's a possibility. And I'm going to introduce that from the beginning. I hope they buy it because it doesn't make sense to start the whole, you know, I want to introduce that idea from the beginning. And I think even we have evidence for that because we have data with other animal models of different muscular dystrophies. So I think I want to do as much shortcut, believe me, as I can without compromising the rigors of what you're doing.
Great. Well, thank you so much for your time today. Thank you for the work you're doing. And we hope to have you back again sometime.
Thanks, June. Thanks everyone for listening to me and for the great questions. And we're working hard. Hopefully we'll get there.
Great. Bye. Thank you.